So welcome everyone to Strait Science. Strait Science is a evening seminar series put on by UAF Northwest Campus in Nome, and also UAF Alaska Sea Grant here in Nome as well. And both UAF, University of Alaska Fairbanks Northwest Campus, and Sea Grant are public servants of the Bering Strait region. And the Bering Strait region is the homeland and waters of the Inupiaq, St. Lawrence Island Yupik, and Yupik peoples. So tonight, kind of a 1-2 punch. If you're reading the Nome Nugget this afternoon, just came out, you'll see that Dwayne Stevenson's presentation last week is on the cover. So well done, NOAA Race Division. And no pressure to our speaker tonight, Dan Cooper, but he is going to give the sort of the second part of the story. So whereas Dwayne Stevenson gave us what had happened during the Northern Bering Sea bottom trawl survey, we're going to hear from Dan Cooper, who is with NOAA Race Division, and that is the Resource Assessment and Conservation Engineering Division of NOAA Fisheries. And these are the serious researchers that are looking at the Bering Sea and especially the Northern Bering Sea with an ecosystem-wide perspective. So he's giving us what happened this summer with the surface trawl survey, but I'm curious because they put a new piece of the puzzle in their research, and that is the surface trawl survey actually also did a little bottom, we'll hear about it, a little work on the bottom, on the seafloor. So we'll hear a little bit more in depth too about what they found on the bottom. So very exciting for us in the region, and we really appreciate NOAA Race Division giving us their time like this. Dan is in Seattle right now, so he's already on a different time zone, so it's late enough, and he's still in the office, so we thank you for that right off the get-go. And Dan Cooper is a research biologist with NOAA Race Division, and he was out on the the Northwest Explorer was the ship you guys were using?
Yes, it is.
Okay, so they, believe it or not, I happened to be taking some pictures of the port the night of Murbach's arrival to the region, and in comes this ship, and I'm thinking, get out of Dodge. What are you doing pulling into the port the night of Murbach? And they were literally meeting up with fish and game research biologists, literally throwing a cooler of samples and getting out of Dodge. And they ended up hiding behind St. Lawrence Island, right near Gamble, during the height of Murbach. So so we give you a shout out. You were with us on that one, and that was a big deal here. With that, I better stop talking and let you take it away, Dan Cooper. Tell us, what did you find on top and all the way down to the bottom?
All right.
Thank
you, Gay. And thanks for organizing this series and allowing us to present. I will say that the work that I'm presenting today is the work of many researchers, and hopefully as I go, I'll remember to attribute, people's work to them. If not, and I forget, many of my slides will have people's name, the people who did the work. And thanks to everybody for attending, taking time out of your evening. But Jim Murphy has led this survey with collaborators from NOAA and ADF&G for 20 years, the surface trawl survey. And much more recently, some others, including me, have added some bottom sampling to the survey. And so tonight, kind of what I hope to tell you is, and we'll go through in that order. At first, I will give some results from the survey that we did in September, 2022, show you the recent results and some long-term trends from the survey for sea surface temperature, zooplankton, and salmon and forage fish. And then I will tell you about the benthic or bottom sampling that we've added to the survey. And I'll tell you why we've added it, what we were doing, and give you some initial results from that work as well. So the Northern Bering Sea Surface Trawl and Ecosystem Survey is the name now. It's been going for 20 years and it's been funded by different initiatives. So the names have changed a few times, but It is a surface trawl and ecosystem survey and has been for 20 years. It's done every year in, or target is the first 3 weeks of September. And the area you can see are these, small black dots on this map of the northern Bering Sea. It goes from Nunivak Island, up to the Bering Strait. And initially, it was a survey to study juvenile salmon in the ocean. And it's acquired other objectives. Now we study forage fish, oceanography, seabirds and marine mammals, which I'm not going to talk about tonight. And then, like I said, we've added some benthic or bottom sampling. So go through, just highlight one result from oceanography. This picture on the left is a CTD, an instrument that we deploy at every station on the survey. And it collects a lot of information about the water properties. The one that I'm going to talk about tonight is temperature. And this figure that you see is sea surface temperature, kind of an average from the survey area during the survey time in August and September over time. And the survey has been going on since I think 2002, 2003. These yellow dots are CTD sea surface, temperatures that were derived from these or measured by these CTDs. And Jim has matched that up with some satellite data shown here by this gray line. And he did that so he could extend the time series back, as long as these satellites have been taking data. And so he goes to the mid 80s. This shows, the annual sea surface temperature, kind of a mean from the survey area at the time of the survey from the mid 80s up until this year. And then this blue line is kind of the average sea surface temperature for all the years. And what you can see is, I think you are very aware that the past several years have been very warm. And this figure shows you that. And then another main point I want you to get out of this figure is that the very last, data point, that's for this year, it was much colder than it had been in more recent years. And in fact, it's even a little bit colder than this long-term average from the mid-80s. All right, the next results I'll tell you about from the surface trawl survey are some plankton trends. At every station also, we deploy this net that you can see in this far left picture. We call it a bongo net. It's very, very fine mesh, and it collects, it collects plankton. And the middle picture here is a scientist, counting the different types of plankton. And then this picture in the oval, is magnified, shown from a microscope of some copepods, one of the primary plankton species, that are feeding a lot of the food web in the northern Bering Sea. And so these figures are some results from the entire time series of the survey, the last 20 years. The upper left panel is the abundance of large copepods every year. And this year there were slightly more large copepods. These are bigger than 2 millimeters. There are slightly more of these than there had been in previous years. The middle panel shows the abundance of small copepods that are less than 2 millimeters. And it's a little bit hard to see in this figure because the top panel and the bottom panel just have numbers, but the middle panel for the small copepods is a log scale. So it looks like there was a little bit of a decrease this year, but because it's a log scale, each one of these numbers, as it goes up, it's up by 10 times. So actually, the small copepods this year declined by an order of magnitude, which means there's only 10% of what there had been in many of the other years. There was a big decrease in the small copepods. Then the bottom panel shows euphausids in the plankton. They're also known as krill Copepods are a little bit, are definitely smaller, but they're an important part of the plankton. Euphausids are also an important part, but generally in the northern Bering Sea, there's a lot lower abundances, although in the past year, they were higher than they had been in other years, and they've been increasing, every year since 2018 in the northern Bering Sea. All right, another piece of gear that is deployed on the surface trawl survey is the surface trawl. You can see a schematic of it in the upper left, and then in the lower left you can see the crew emptying the catch into our sorting table. But it's a large trawl. This other picture is a picture that was taken from a drone, and it's taken of the trawl fishing and the boat heading away from us. And these things that you see trailing along in the water are the floats. Again, it's a surface trawl. We're trying to catch the juvenile salmon and other animals that are in the surface water. It's a wide trawl. It's about 50 meters wide, but it's used because it does a good job of catching juvenile salmon and forage fish. And so next I'm going to show you some of the species that we catch in this trawl, the results from that from this year, and trends from the survey over the whole survey time period. The first animals that we're going to talk about, the first fish, are herring and capelin. On the left is a figure that shows herring abundance over time on the survey. There's dark blue points, and those are from the northern Bering Sea. The light blue are from the southeast Bering Sea. But you can see for the northern Bering Sea, in the most recent years of the survey, herring numbers have been very low compared to what they were in the past. The majority of herring in the northern Bering Sea are juvenile, and peaks in these, when there's high peaks in the numbers, that could represent good recruitment events that are later seen, in herring as age fours in the Togiak fishery. But again, the last few years, herring numbers have been low. Then the panel on the right is capelin. You can see that capelin, I should say, is another forage fish species like herring. It's very important to the food web. And you can see that capelin numbers were higher this past year than they were the year before, but also they're kind of low by historical standards. And large capelin biomass estimate years tend to be in years of cooler water and larger cold pools. So maybe that's why there was an increase. I should say that this work was done by Alex Andrews and some others. All right, the next species that we catch in the surface trawl that I'm going to talk about is a juvenile Chinook salmon. This figure on the left is an abundance index of juvenile Chinook salmon caught in the northern Bering Sea survey. It's a mixed stock abundance index, meaning that fish, these juveniles, could have come from the Yukon River, the Kuskokwim River, the Unalakleet River, and others. So it's, that's right now the data show the fish coming from all these different rivers, and they're all totaled in here. The figure on the right, you see this map, shows the distribution of where the juvenile Chinook fish were caught in 2022, just this last year. You can see that they were primarily distributed in the northern part of the surface area in Norton Sound or adjacent to Norton Sound. All right, so I said this was a mixed stock. These fish are coming from different rivers, but on average, over 80% of these juvenile Chinook originate in the Yukon River. There's genetic ID work that's being done by ADF&G, and they will be able to apportion out, these fish that we caught into what river they were coming from. But on average, usually about 80% of the juvenile Chinook salmon caught on the survey originate in the Yukon River. And the juvenile abundance, this index, how many fish there were as juveniles in the ocean, is highly correlated to later adult returns on the Yukon River, which means a couple things. One, it means that whatever makes a good year class or a bad year class or a bad year class for the salmon usually happens before the survey. So it's happening at early life stages. And that also means that whatever is caught on the survey provides a reasonably clear expectation for what's going to return to the Yukon River. And so if you look at this series of abundance in, this past year, it was rather low. It had been declined, and the numbers have kind of steadily declined since about 2013. And so, this work done by Jim Murphy and others would suggest that the declines of Yukon River Chinook are going to continue. Then the next salmon species I'm going to show you is juvenile pink salmon caught in the survey. Again, this shows the mixed stock abundance index from the survey, how many were caught this year and all the other years of the survey. And again, you can see there was a large decline this year and last year. On the right, you can see the distribution of where we caught these. And this distribution is not typical. Most of the time in previous years, this year, more fish were caught up here in the north near the Bering Strait than is typical. Again, this is a mixed stock. These pinks are coming from many places, but the index here of what's caught on the survey is highly correlated to returns in the Yukon River and in Norton Sound. So these low numbers mean that there are probably, they're expecting poor returns to Norton Sound and the Yukon River in 2023. This is work that Ed Farley is leading. And then the last salmon species that I'll talk about is fall Yukon chum juveniles. Again, this index shows how many fall Yukon chum juveniles that we expect that we caught in the survey. You can see that for this year, the numbers were very low for the average. This dashed line, it represents the average of the time series. You can see that some years the numbers are way above average and some years are well below average, and this year was the lowest on record. The map on the right shows where we caught these chum salmon. And this is also not typical. Normally chum salmon are caught across the survey grid, but in this past year, they were caught in the nearshore areas, and also in 2021, they were caught in the nearshore areas. So something is changing with their distribution. And the salmon biologists do not know why this pattern is occurring. Typically, when there's the more chum salmon juveniles that are caught on this survey, means there's more fall chum returning to the Yukon a few years later. But during this last marine heat wave, that didn't hold true. Some of these years with big, abundance out in the ocean did not lead to big returns. And so that is still kind of under investigation. And the salmon biologists, which is led by Sabrina Garcia from ADF&G, are continuing to monitor the chum salmon abundance in the northern Bering Sea and continuing to explore ways of how to use the abundance to, as a prediction of future run sizes in the Yukon River. And then the last thing that we're going to talk about from the surface trawl survey is we caught a salmon shark. It's shown here this year. It's a female salmon shark. We tagged it. Sabrina Garcia, again from Alaska Department of Fish and Game, has led efforts in the past several years to put satellite tags on salmon sharks in the northern Bering Sea. And if you look at the dorsal fin, you can see that we just put on a satellite tag. These tags, every time the shark surfaces enough to poke its fin out of the water, the tag beams its location to a satellite, which then Sabrina collects those data. So this was tagged around on September 11th. It hung around, this map shows where we tagged it. And it was in the northern Bering Sea until the end of September, and it went to the Aleutian Islands. Sabrina Garcia is going to be giving a straight science talk later this winter about all of our salmon shark tagging results, and that should be very interesting. So be on the lookout for that. All right, now I'm going to shift gears a little bit and move into the bottom sampling that we've added to the talk. And again, I'm going to talk about why did we add it, how are we doing the bottom sampling, and what are our initial results. And so the reasons we added bottom sampling, one is to monitor small animals on the seafloor. And these, could be small animals that are always small as adults, like shrimp that are part of the food web, an important part of the food web, or animals that are small, like young of the year, Pacific cod, or snow crab, that we don't know as much about. For instance, we know from work from Dwayne Stevenson and the bottom trawl survey that there are a lot of Pacific cod that have moved into the northern Bering Sea in recent warm years. But one thing we don't know as much about is these small Pacific cod. These ones are about the size of your finger, and they're, the first year they've been alive. We don't know as much about if they are living in the northern Bering Sea and if cod have started to reproduce and the whole life cycle is taking place in the northern Bering Sea. So that's one thing we're trying to study. Another thing we're trying to study about these small animals is their body condition, how much fat they've stored, because this is important to overwinter survival, and also their quality as prey items. Another reason we've added bottom sampling or benthic sampling is to get more samples for harmful algal blooms and the efforts that are underway to monitor those in the region. And also, we're trying to study ecosystem change with the warming ocean using these small animals. And I'll show you next an example of a project that we have going on right now with juvenile snow crab. When I say juvenile snow crab, here's a picture of them. I'm talking about ones that are about 15 millimeters or less in size. So it's about the width of your little finger. But they're so slow growing that they're about 2 years old, we think, at that age, but that's the size we're studying. And so this project is being led by Louise Koopman, also of NOAA. And her idea is to study how the warming ocean and reduced sea ice is impacting these smallest crab. And they could be being impacted by some direct effects. We know that as the water warms up, the crab are able to grow faster, these small crabs. But the trade-off there is that they might store less fat for winter, and there might be lower survival rates of these smallest crab. A warming ocean could also impact these crab by some indirect ways because of the food web. For instance, we know that different types of phytoplankton or algae are associated with sea ice or open water. And the different types of phytoplankton, they lead to different amounts and different types of food falling to the seafloor that feed the whole benthic food web. And the idea, the hypothesis, is that less sea ice could mean less food, and less food with less fat in it, or less fatty food, falls to the seafloor. And so crab could be impacted, and fewer young crab could survive, leading to declines in the population, or the crab population could move. So to study these ideas, we're measuring the distribution and abundance of the smallest crab. We're measuring fat levels in the crab and the sediments, and we're also measuring the types of fat, what kind of plankton they came from in the crab and in the sediment, where it's fallen to the seafloor. All right, so the tools that we're using, that we've added to the survey in order to sample these small animals in the sediment, one is a small mesh trawl that we added 2 years ago in 2021. And then we've also added a benthic grab in 2022. And so one question that I think many people have is how is what we're doing to sample these bottom animals different from what the bottom trawl survey is doing. Many of you have seen um, people from the bottom trawl survey explain their results, and they do a wonderful survey. Dwayne Stevenson just gave a Strait Science talk last week and also earlier in the year. And before that, Lyle Britt was in charge of that group and gave many talks explaining what they're doing. And so I just want to show how what we're doing is different from what they're doing. This picture on the left is the bottom trawl survey net, the trawl. It's strung up at our net shed. What you can see is it's a pretty big net. You can see those people behind there. And the survey net we're using is this one on the right. If I make them about the same size, you can see that ours has a much smaller mouth opening. It's about a 6 foot wide. This is the only area that we're fishing. And because it's so small, many large fish avoid our net, and it's not very good at surveying most adult fish. But what our net is good at, if I zoom in and show you the bottom trawl survey net again on the left and ours on the right, is that ours has a much smaller mesh. And what it's good at is retaining these small animals, these small fish and other animals that we're trying to study. So aside from that small mesh trawl, we've also added a benthic grab sampling. This picture on the left is our benthic grab. We deploy it down to the bottom, and it scoops up and returns some sediment to us. And from that, this middle picture shows it's got a little door on top, so we can open that door and we can see the sediment that had been just right on the top of the seafloor. And from that, we take samples for the crab and other projects to see what type of food fell to the seafloor. And we also take samples just to see what kind of sediment type is there, because different animals prefer different sediment types. And we also, after we're done with that, we also take HAB samples from the sediment. And then after that, we sieve the remainder of the sediment that we catch, and you can see these worms on the top and clams that we sometimes catch, and we save those also for harmful algal bloom sampling. So to present some of the initial results of our new gears, at first I'm going to talk about the trawl catch. And I've organized first the invertebrates that we catch kind of in decreasing weight, total weight from the survey in this past year. We catch a lot more types of animals than this. We catch I think the list was 55 invertebrates and 37 fishes, and it's 55 either species or groups of species that are more difficult to tell apart. And I'll kind of go through and use this, to guide the species that I talk about. The first one is sand dollars. It's been the highest weight species that we catch in the last 2 years. Here's a picture of one of our catch. They can be quite large. And I'm showing you this mostly because, it's so large. But they're only from a few different areas. And I think the number of things that eat sand dollars is pretty limited. So that's all I'm going to say about sand dollars, although it seems to be a big part of the ecosystem in some places. After sand dollars, the next thing we catch, the most biomass of, is purple orange sea star. And there's a picture of that here. And then I've also highlighted several other sea stars. On this picture down on the right is the polar six-rayed star, or Leptosterius polaris. And then there's some others we catch. We catch a high biomass of these sea stars. And here's just some distribution of two of them, the polar six-rayed star last year, and then the purple orange sea star of where we catch them. That's a very common species in the near shore. After that, a species that our net does, I think, a more unique job of catching and determining abundance of are some shrimp species. It turns out in both years, some of our bigger catches, some of the animals have been different shrimp species. Ridged crangon, seven-spine bay shrimp are another crangon species, and then Argus spa is a genus of shrimp that we're trying to learn more about how to tell them apart, but right now we we lump them as a genus. I think these ones are important because, you know, they're species that are small. They look like they're good prey items and part of the food web, and so we're starting to study those more. Here's a humpy shrimp, one species of shrimp we catch that's more of the offshore part of our survey area, and then ridged crangon, which is the highest biomass shrimp species we catch, and seems to be a good study animal for us, for abundance and for special projects to look at how their fat content also changes like the crab. And then the last one I'll talk about are the snow crab. These are, you know, one of the species that we're working with right now. And the snow crab in 2021, these are, again, just the smallest ones, less than 15 millimeters, about the width of your pinky. We caught them kind of in two areas, offshore in the southern part of the grid and then the northern part of the grid. And we caught them at a few less stations in this past year. All right, now I'll go through some of the top fish species that we catch in the trawl. You notice I'm doing this by number rather than weight because we catch a few skates and a few adult sculpin that would be much better surveyed by the bottom trawl survey, and they would dominate my catch by weight. So some of the species we're interested in are, you know, have higher numbers, like yellowfin sole. We catch them down to about 3 centimeters. So these are juveniles of the species. You can see that in both years, these fish were caught in the near shore areas, but that's something, the smallest of these fish that our net would do a good job of monitoring. After that, the highest number of fish we catch are these flatfish unidentified. And they're unidentified because I left them in this picture. You can see them here. These fish have recently settled to the bottom. We have a mixture of species that we're trying to deal with. A lot of these have characters still of the larvae. They're clear and they've just settled on the bottom. So we've taken these fish to the laboratory and we hope to have taxonomists help us tell exactly which species we have. So I don't have a lot of other data on them, but that is something that we also catch a lot with our net. Another species that I'll talk about a little more is saffron cod. Some people know these as tom cod. They're an important subsistence fish. What I'm going to talk about is just the smallest ones. These are mostly from about 6 to 10 centimeters. So again, about as long as an adult's finger. And our small mesh net has caught these in both years, 2021, this map on the left, and 2022 on the right, in these nearshore areas. So again, that's another species and another, you know, young of the year species that we should be able to monitor with this net long term. After that, we also catch a large variety of small fish. Snake prickleback are these ones. Sculpin unidentified. Again, they're unidentified because many of them are only a couple centimeters long, and so we've frozen them and brought them back. And you know, we catch a wide variety of very small fish. And one species that did not make the top list, but I think is important again, is Pacific cod. Again, these are age 0, is about as long as your finger. In 2021, a warm year, we caught a few of these, not many, a very small number in the northern Bering Sea. And then just in this past year, we didn't catch any. And so I said that, you know, we would like to monitor reproduction of Pacific cod in the northern Bering Sea, and we don't see a lot of reproduction of the cod being successful. Maybe it's in other areas, but we have no evidence that that is happening. And then finally, I'll show you a slide of the samples that we gave to the people doing HABS work. Here's a picture of Kathy LaFay and her team, and they provided this map of where we caught clams and worms in our sediment grab, and we gave them actually some sediment from all these stations. And in the clams and worms, they're testing for some of the actual toxins and the cysts, the alexandrium that's making the sax toxin. And in the sediment, they're looking for the cysts, these things that might lie dormant on the seafloor. And so our work has added to the amount of HAB sampling going on in the area. All right. There's a lot of people to thank for this work. I will say we're very grateful to Jennifer Bell from ADF&G. As Gay said, we finished our survey right before Merbach hit, and we dumped our samples, and poor Jennifer had to have our samples. Some of them didn't get sent right away on Alaska Airlines. They got rejected, and she had to take them back and keep them frozen for us all this time when this horrible storm was going on. So we're very grateful to Jennifer Bell for her help, for getting our samples where they need to go and keeping them frozen. It saved, there's been months and months of work done on this crab project, and those are the ones that she saved. So thank you, Jen. Also, some of the people that were doing the work on the ship, some of the science crew are Jenna Barrett, Josh Clark, Samantha Decker, Elizabeth Lee, and Alexa Pinchuk. And those are just the ones from the past few years. There's been, countless scientists going back 20 years, and the captain and crew of the Northwest Explorer, the boat we've used for the past few years. Mike Levine from NOAA helped us with some data, and Gay Sheffield helped us put on this talk. Oh, I should mention Diana from the Nugget let me put this picture of Merbach hitting town. So then here are pictures of many of the people that have provided the work that I just talked about, and I can answer any questions now that anybody might have.
All right. Well, thank you so much, Dan Cooper. And yeah, that picture of Nome, that was post hitting. That was sort of the day after when things were slowing down. So you kind of get the idea of that. But thank you. And now we'll do questions. But while people are thinking of their questions, this is a good time to throw a little love to Dan Cooper and the race division in the chat box. It's not easy to be a straight science speaker. And so we thank Dan Cooper, especially since he's on a different time zone. So it's, it's past 8 o'clock for him. And he's still in the office. So, yeah, you can just say stop share, I think, and it'll hit that red button, and then your face will get bigger and we won't have to see me twice. But thanks. And so let's see, does anyone have any burning questions about what we just learned? You're getting some claps and some nice kudos in the chat box, but all right, nothing burning. So I have a question, and that is, did you not get any king crab in your little net?
Well, we've caught a few king crab.
Okay.
But again, they're big enough that I think if anybody's concerned about, if the best data source is going to be from other sources, the Bottom Trawl Survey, or I think there's a Norton Trawl.
Yep, fishing game, Norton Sound. Yep.
Yeah, and so we've caught like 2. I could, it's okay. A lucky shot if we catch one of those.
Okay. All right.
I have a question.
Yeah. Okay.
Go for it. Yep.
Not Megan Gannon, so this isn't coming from the Nugget, but
okay.
So on, I guess it was slide 10 or so when you were looking at the juvenile salmon. If I look back in the time series a little bit, at I guess it was 2013 to 2014 transition or somewhere like that, you had years where, so you commented on, years when there was a big juvenile output, and then the next year, this past year, there was not so many juveniles. That seems, conditions out at sea weren't ideal for them to grow up. But then you have years where there was low output, and then the following year, high output from these rivers. So there was a lot of juveniles coming out, which implies that either, I guess for me, that would imply possibly 2 things. There's more favorable conditions upstream, that is, they're able to breed and reproduce more and produce more juveniles, this lower amount that's coming up, or there's some missing samples, because this is a large area. So I'm wondering if these might just be errors in the sampling, or whether it is something that's causing all of a sudden a big bloom of juveniles or, big increase when the previous year was so low.
Well, do you remember which species you were looking at, or is it just in general
Just in general, just when you see a
Okay.
bar that's like a low amount of juveniles, and then the following year a very high amount.
Yeah, I mean, I
What breaks
think,
the pattern? Yeah.
well, I think, for the species where there's a good correlation between what's seen in the survey and then what later returns to the river to spawn, for those species, like something is still making good year classes and bad year classes. But for those ones, I think is Chinook and pinks, that something happening before the fish get to the survey. So something, between when they're spawned and they make it out into the ocean, something could be making good year classes and bad year classes. That's, it could be something that's happening in the freshwater before they get to the marine environment, or when they first enter the marine environment and before September, when we do the survey. But Jim Murphy, I'm sure, is on the line, and he could probably tell you more about that than I could.
Jim Murphy, you got anything to add?
Well, hi, can you see me?
Yep.
Okay,
Right.
well, I can see myself, so. No, I think Dan's done a good job. I think maybe if you start to think about pink salmon itself, because they have this short life cycle, there may be, some dynamics there that would be different than Chinook, which have a much longer life cycle. So there could be some feedback with, the abundance of spawners that are tied to, the subsequent juvenile year. But in general, it's really what's controlling your class strength can vary from year to year. So just because you have, poor survival or poor juvenile abundance one year doesn't necessarily imply that you're going to have a similar poor abundance in the following year, because the processes that are determining your class strength can vary quite a bit from one year to the next. Hopefully I didn't just confuse everyone, but
Did that get the question answered?
Yeah, so I guess it did. The second part of my question was just whether it could be a sampling issue, because this is a very large area, but it looks like, it's well placed. The surveys, how consistent are the fish always coming to the same place, or are you just at the mouth of the river? So, that would be I guess the ideal sampling location, right at the mouth of the river. But you don't think there's any, short my question, you don't think there's any error in the sampling that could be causing this? It really is the true signal of the fish.
Like some kind of error where they're swimming outside of the survey area?
Yeah, or you're missing, parts of the survey area, fish are getting away.
Yeah.
It looks like it's pretty well covered.
Well, I mean, I think in years when that happened, maybe there would be, you could see big abundances right on the edge. There might be some that you missed if that happened, on the edge of the survey. I think Jim has tried to make the survey so that the outer stations have really low abundances to make sure he's got it. And again, Jim would be better able to answer how well, if there's sometime in the 20-year time series, if he thinks that sometimes it hasn't, covered the species well or not.
I have a salmon question. Are you feel you got your answer? Okay. Oh, Charlie, go ahead. I see your hand is up. You go before me.
Yeah, well, I apologize. I missed part of the presentation, but these last questions about the cause for the low numbers apparently happening before the trawl survey. One of the strong correlations we found on shore was that the coho and pink salmon abundance, strong even years, weak odd years, and there's no doubt that the coho are big predators of the pink salmon. And then when I was trying to raise fish, I inadvertently had some coho and chum hatch at the same time, And so I thought that was cool, and I stuck them in a living stream aquarium, and I put a barrier between the two stocks, but it wasn't effective. And what I found was that the cohos of the same size would gang up on a chum salmon and eat him. And in about a week's time, I had no more chum salmon. So those cohos are little piranhas. And I would think that might be they're kind of a recent immigrant to this region, and they might be part of the issue.
Is the coho?
The cohos as predators, and the juvenile, and when they go to sea, coho smolt, pink fry.
So there's no question associated with that, Charlie. You're just passing along that information.
Well, yeah, I mean, I understood that the cause for the weak years was apparently nearer shore or even in fresh water, and that's one of the things I don't think people have looked at. So, yeah.
Near shore.
Comment.
Yeah, I was going to ask about what the working hypothesis. I mean, it was really noticeable, but no chum, again. And so what is there a working hypothesis on why they may have moved? And I see you, Dean, and we do also have a question in the chat. I see that. And Jamal. Is there?
Well, again, for the salmon questions on why chum, abundances are so low, I would defer to Sabrina if she's on the call. She
is not, but
or Jim Murphy. They're
Jim Murphy,
salmon biologists.
You're up again, Jim. Lots of salmon questions.
Well, or maybe we could go to Jamal.
All right, Jamal. He's got a question and he's going to try to answer.
I'm the one trying to ask questions, not give answers.
All right.
Okay, I will then. I think there's a couple of questions related to chum, and part of the question is why we're not seeing the returns to the river when we do see good juvenile abundance, and that points to the fact that I think we're seeing this issue during these marine heat waves where the juveniles are not surviving. So that could be in part because some of the warming of the northern Bering Sea is reducing their condition, and so the juveniles may be entering the winter at poorer condition. But I think the other part of that is that the Western Alaska chum salmon spend most of their life in the Gulf of Alaska, so they'll migrate south and in the Gulf of Alaska. So it's really about some of the conditions that are occurring offshore, and at that point we think that that's also a very important part of the story as to why we're seeing issues with chum salmon in the Yukon and Northern Sound and Western Alaska in general. So I think right now that's kind of the those are some of the key ecological points that tied to the poor survival of chum salmon.
All right, thank you, Jim. I guess that's the answer at this time. So in the chat, and then Dean, it looks like he got knocked off. No, Dean, you're still there. But Landry Price has in the chat, it's been patiently waiting. Is there a graph that can be integrated into the juvenile abundance of both Chinook/chum with the number of prior year abundance of spawners? Is that something that could be done in the future?
I think that could be done in the future, and I don't know. Perhaps I imagine that's already been done. I hate to keep kicking it to Jim, but I imagine that he would know if that has been done already, what can be done.
Yeah, you're right, Dan. We've done that for Chinook. So we characterize Chinook in terms of the number of juveniles per spawner. So that's, but Dan's just not showing that. And in theory, we could do this for chum salmon, but no, know if that's been done yet. But we have done it for Chinook.
All right.
Thanks.
And-
Oh,
and
can I
Jim-
just respond real quick to that?
Yeah.
I was just gonna say, thanks, Jim. What is there a real positive correlation between the number of spawners and what you see in the juvenile abundance in the survey?
No, no, that there's a lot of variation in the number of juveniles produced per spawner.
And there's a comment to follow. Jamal put in chum salmon captured in the Gulf of Alaska during winter this year. So that would be the January, February sort of thing. Okay. During winter this year, as part of the International Year of the Salmon Survey, were in poor condition. So good to know. I mean, that's interesting for us up here. It was very noticeable. Second year in a row, didn't put out a net at our house. All right, Dean, I don't know if you still have your question. Your hand had been up.
I do. And I guess, first of all, Dan, thanks for the presentation. Really enjoyed it. Really appreciate the effort you guys put into this. My question is, all the distributions that you show are cyclic and some almost decadal in their response. So how do you separate, like, if this was a bad year from all of the other years? I mean, it may be the low point in a 10-year cycle. How do you interpret that kind of data? Okay.
Well, I think there's
I didn't mean to blindside you with that, but let me give you an example. You have reduced sea ice. You have a reduced cold pool one year. You have increased movement of adult Pacific cod and pollock moving north, increased predation. All of this impacts the benthos. These things are tremendous to me, and I just don't see how you put it into context. I guess that's what I'm asking.
I think it's difficult and something we're working with. I think for like a lot of the species, like I showed trends of capelin abundance, for instance. And, it's like for us, for species that live more than the longer species live, the more that there can be, some sort of a buildup of conditions. Good conditions will slowly build up the species, and bad conditions will slowly, degrade the species. But so it's difficult, especially for a long-lived species.
Okay. Yeah, understood.
it is, and for you're right, the ecosystem can change over time, and we're back to normal, kind of, well, not normal, but we're back to an average temperature, but everything doesn't just swing back into place immediately or something like that. But I think that's something we're all working with.
Thanks.
Yeah, and that's the big question. And I appreciate that, Dean, and also your response, Dan. I mean, we are seeing the whole ecosystem here, right on up to the marine mammals, seabirds, it's incredible what we're witness to right now in the last 10 years, right? 20 years. But really that blowout of the cold pool in 2018, I mean, everything is banging, all the things that are out of their norm are just banging into each other, and then they've got an effect, and it's really extraordinary. So we're glad you guys are up here doing that and putting the northern. I know you guys have been surface trawl for a long time, and we're really grateful you're letting us know what you find. And but we're really grateful for all those fish surveys and the bottom trawl survey as well. That's kind of new to the region, but boy, we need help here in figuring out this giant puzzle. So thanks for that. So Jamal, you have a question.
Thanks, Kay. I just want to say thank you to Dan and Jim and everyone that's been part of the Northern Bering Sea Surface Trawl Survey. Huge effort. Thanks for showing up tonight. Thanks for everyone that's on the call tonight from all over the state and contributing. Really appreciate that. My question is more sort of a question to all of us, even to myself, and definitely willing to step up and try to answer some of these hard questions and not throw them, to Jim's way and other people's way. But we have like Marty and Charlie and Jim and all these wonderful people, and even folks who I might not even know. I see their names here, who could be elders or very knowledgeable folks that know about salmon ecology and are actually on the ground and actually witnessing and seeing a lot of the stuff that we scientists aren't privileged to see. So I really appreciate everyone's participation tonight. My question was kind of focused around salmon as well, around both Chinook and chum. And so some of the things that were presented tonight that kind of caught my attention were, Chinook were distributed pretty far north this year, even though it was a cooler than normal year. Same with pinks. Typically, I think in the past, we've seen these more northernward distributions in warm years, but now we have a cool year and they're still further north. And so that's kind of a stick it in my mind and like wondering what's going on there. But sort of the bigger question is, given that we're witnessing all these changes and things are, changing very rapidly, and we're seeing a lot of things that we're not expecting to see that don't quite make sense. As far as like what we're seeing with juvenile salmon, I wonder if folks have ideas about what new questions or new sort of focal areas we should be looking at in the ocean post-juvenile ecology, maybe, one ocean year perhaps, or beyond that might be able to help us answer some of these questions, because it seems like there's been sort of a decoupling between sort of like year class strength being set before, for example, juvenile Chinook hit the ocean, and some of our time series kind of breaking down. And so there's something going on that I feel like we need to kind of get a grasp on and understand that's out in the ocean. For juvenile chum, we know a lot of that mortality is happening for them out in the ocean. And so just curious to know if people have ideas on maybe ideas for expanding some of the things we're looking at, ways to do that, what we should be looking at to try to get an idea on what some of these impacts on these important stocks might be. So just sort of an open question on what we could recommend based on what we know here to others. And of course, there's a lot of interest right now, especially with like our state legislatures, our federal representatives, in trying to address, bycatch issues, but then also ecology issues with warming the Bering Sea. And so I think right now we're poised to take advantage of some support, but I think people are really looking for direction, and for folks like us that might have, a little bit more knowledge to kind of point them in those directions on what we need to kind of focus on going forward to help understand these puzzles that we're seeing right now.
All right, that's a question to all of us. Thanks, Jamal. Anyone want to take a crack at that? Dean, I don't know if your hand's raised to answer Jamal's question or you have another question.
It's another question, so
okay.
I'll let
Do you have
go.
anything that NOAA should be looking at to answer that first one?
My question is, with all the sampling that they're taking, and I know they're overwhelmed with what they're doing, do they take stomach contents and analyze that? For example, if you look at cod, I know that cod will eat sea urchins. Do you see any of that taking place? And, I know you guys are taxed to the limit in what you're analyzing, and I do appreciate it, but thanks.
Well, I can answer that. On the surface trawl survey, we do take stomachs from many of the different fish, all the salmon, I believe, and a lot of the forage fish. We take salmon. from. And then the bottom trawl survey, which I've talked about, takes stomachs from, I don't know which exact species, I'm sure it's cod. But they take from a subset of the species, they take stomachs of the adult fish. And so, yes, that is being studied. Specifically, I don't know about sea urchins in cod, but that is being studied by there's a group that studies stomach content of the adult fish. And then people that work with Jamal and Jim also are doing stomach contents of the fish caught on the surface trawls.
Thanks, Dan. Appreciate it.
Thanks, Dean. And Dwayne says we take stomach samples from cod and pollock on the bottom trawl survey as well. Anyone have an answer for or want to take a stab at Jamal's question? Charlie?
Yeah, I over the years I've seen that. So I've lived in Norton Sound for most of my life, and years ago there was a much stronger alignment with when the sea ice went out and when the river ice went out, and that was partially because there was a greater shorefast ice component or a bigger shelf, and it was also has to do with the change in the climate that we started to experience as far back as 1990. And so beginning in 1990, we noticed that the Norton Sound king crab were no longer kept out of eastern Norton Sound because it was so fresh. When the king crab fishery first opened, you couldn't keep crab in your hold east of Cape Nome. But then within 10 years later, you could keep crab clear over by Golovin Bay. The salinity was changing, and that was in August. But in the early summer, it was even more pronounced, much fresher water. Coincident with that salinity change, we saw the Capeland move further north and out of Norton Sound. They were very, very, very dense and common in the 80s, and by the late 90s, they were very few. So I think one of the things to look at is late springs and early springs and how fresh is the lens of seawater at the river mouths when the salmon go out to sea. If it's relatively fresh, the salmon can disperse and they aren't concentrated for the predators to hammer them. If it's a real sharp demarcation with fresh and then salt, the predators have a field day, both avian and fish. So I think that salinity issue, and which has to do with the ice out on the ocean, is a big deal. And I would suggest you start looking at salinity.
Thanks, Charlie.
Thank you, Charlie.
I would add one if no one else is going to jump in on this. So for the ecosystem, and maybe it's just my ignorance, which, you know, is pretty profound, but sea ice, algae is the primary production for a lot of the krills. And then it also provides a subsidy for our already rich waters, both the Onondaga Current and the Alaska Coastal Current. And the sea ice used to be fairly extensive. And so we'd have animals, you know, there's just a ton of food. We're rich water, but we were even richer. And when that ice algae would let go, right, you get those mats. I mean, you could even kill off clam beds. It would just be too much food and bury things in food. And that is why I've always been taught we are the premier breadbasket, and we're pumping we're feeding everything 12 months of the year, whether they're coming up from the north or coming up from the south or coming down from the north. I mean, 12 months of the year, we are feeding massive amounts of birds, seals, fish, crab, you name it, in the northern Bering Sea. So we've lost a tremendous amount of ice. You can measure it, right, with a satellite, and you don't need to worry about the border. You can just get a nice what amount of primary production have we lost on an annual basis? Because you could then maybe on the back of a napkin say, all right, well, there's a lot less for the krill, or there's a lot less for clams. And then on the top of it, you can throw in that the water's getting warmer. So then you're jacking up the metabolism of all these cold-blooded things like crab and whatever, fish. And so their metabolism's getting cranked up. And if I remember right, you got to eat more. So you're sort of thinking, God, I got to eat more. I'm a fish or a crab. I got to eat more. And somebody turned off all the primary production. And so I just keep hearing things are hungry all over. And on top of it, you drop the barrier and you add in large predatory commercially viable fish from the south that want to get in there, and they're jacked up on temperature too. So they're hungry, and there's our little fish, and our little fish are like, we need to eat too. And then you've got, I mean, is that something that you could kind of look at, the ice algae, just back of a napkin? What have we lost primary production wise to set off? Because I am dumbfounded how hungry everything is, honestly, or that they're physically getting smaller, or that we just don't have as much all over. And so how do you pull that off? It's like the carrying capacity of the northern Bering Sea. I could be wrong here, but has that been throttled down hard? And then you add temperature, so the pressure's to eat more, so you need, you know what I'm saying? And then you've got mayhem going on under the surface because you've got cod showing up, big cod, and you've got our little capelin, and everybody's just trying to make a living and get something to eat. And it is that helpful at all? Kind of starting with the ice algae, and sort of how do these things respond when you throw all those things at it?
I mean, I think you're right. Those are really huge issues right now. All I can say is that, you know, those are the types of issues that we're trying to study right now, specifically about the amount of primary production.
Okay, so someone's doing that.
Well, I know that people are working on the amount of primary production, and they're trying to quantify that with production experiments when we've been to sea. I don't know the specific numbers of,
but not in the water column. I'm talking about up against the underside of the ice.
Yeah, I mean, I agree that it's important. I don't know what kind of numbers, if anyone's quantified that or not.
Okay.
I just don't know.
Anyway, you were asking, I'd throw that in the mix. The other thing I would say is get your NOAA Office of International Affairs talking to whatever they have, whatever rebot it is, Chakot rebot or the Kamchat rebot, whatever the fisheries, federal fishery guys are over on the Western Bering. I don't know how you guys can't do it, but NOAA has an Office of International Affairs, and somehow there has to be some way to talk. So that would be lovely to know what's going on over there. And I know that can't be put on you guys, but maybe your international affairs seems like that's right up their alley, but what do I know? There are problems there. And then lastly, what would be great is, like, can you forecast what we should expect with, I mean, not just the salmon, but on an ecosystem level, just so we can sort of give you some feedback. And, if you tell us, hey, we expect to see buckets of this, because this region, everyone here is utilizing comprehensively the marine ecosystem, right? It's not just about commercial fish. We're heavily in subsistence, and that includes right on up the food chain, and everybody's utilizing all different manner of things. And so you might get more information if you sort of give us what you think might happen. And then we could kind of have a forecast to say, well, that actually, from my perspective, I didn't see that happen. I saw this happen. And it might be a nice way to get, check on some of the things that you think might be happening.
To check on our ideas, to kind of ground truth them.
Yeah, well, you've got a lot of eyes out here.
Yeah.
And that way we'd have something to say, like, well, all right, they think this is going to happen. I'd be curious to see what sort of a preview of what you think's going to happen going into a year, right or wrong. Everyone knows you wouldn't be, it's not magic. It's just what you best can think of right now. Anyway, that's it. All right. So, any other questions for Dan Cooper, or at this point, Jim Murphy, because he had to take on a bunch of the questions. Dan, you did a great job with the delivery and answering questions as well. And I have an old dog that is barking in the background, needing to probably take a tour outside. So I apologize for that because he is downstairs and I can't get to him. But with that, any other questions or comments for Dan Cooper? If not, Dan, if you want to stay on for just a sec, I know you're late, but it would be good just to talk to you for 1 sec.
Sure.
And for everyone else, our next Strait Science will be on the 15th. It's going to be on a Tuesday, and it's going to be live from Sekuliak. They're going to be calling in, although the weather may be something to see for them up there. They're going to be calling in and talking about the work they're doing currently up in the Beaufort and Chukchi Seas. And that is, they're looking at the fate of the ocean water as it goes barreling north through the Bering Strait. And they're also, I believe, going to have some information. They are sampling for harmful algal bloom materials as well up there. So that'll be live from Sekuliak on Tuesday night at 6:30. And with that, thank you all for coming, everyone. Have a good night.